Hello and welcome to our presentation on the evolutionary enlargement of hominoid cerebellar surface area relative to the neocortex. For all future reference, hominoidea refers to all apes and humans unless specified otherwise. As you can see, this project is highly collaborative and consists of individuals from a range of disciplines and backgrounds. Please enjoy.
The cerebellum is located under the occipital lobe and posterior to the pons and the fourth ventricle. The cerebellum is known for its motor functions, coordination and dexterity, even having its own homunculus, and for its cognitive functions, attention, working memory, allocation, multitasking proficiency, and emotions.
Now, recent works have indicated that the hominoid cerebellum has undergone massive volumetric expansion relative to the neocortex. Each figure illustrates a cerebellar hemispheric grade shift between apes and monkeys. In other words, apes have disproportionately larger cerebella than expected of an equivalent monkey neocortex. Human cerebella are less relatively voluminous, 11% of the neocortex versus 13.4% in other apes. Old and newborn monkey values sit at a lesser 9.8 and 9.2%. This disparity is made clearer between human and macaque cerebellar surface areas, 78 versus 33% of their respective neocortices. And so our primary research question is whether human cerebellar surface area is markedly variant from other apes. This could aid understanding of human-associated behaviors such as advanced language and tool use, and how Purkinje cell diffusion through larger surface areas may enhance cognition. We hypothesize that ape surface areas will be larger than in monkeys, as with volume. We also question whether the cerebellar surface area of monkeys remains in line with volumetric findings, and whether cerebellar surface area findings garner insight into the evolution of cognition. More specifically, our aim is to reconstruct the timeline of cerebellar growth by investigating cerebellar surface area size in a range of primates.
To fulfill our aims, we utilised high-resolution flatbed scans scanned by Carol MacLeod of histological coronal sections. These were acquired from the C&O Vogt Institute of Brain Research in the Stefan collection and stained with Creswell violet for greater differentiation of microstructures. To segment scans, we used MicroDraw vectorial annotation software to delineate molecular layer boundaries of the cerebellum and the peel surface of the cerebrum. This facilitated annotation, relocation, combination, and refinement of regions. In the images below, you can see differences between the gorilla, spider monkey, and orangutan sections. Cortical boundaries were annotated according to the granular cell layer beneath them. Granular layer structures, the darker regions of figures, were observed given uncertainty or damage to the histological section, such as separation artifacts. Here you can see the annotations in action. Using every 20th section from each brain amounted to around 100 sections of cerebellum and around 100 sections of cerebrum to annotate. To avoid confounding cerebellar measurements, the pons in each scan was segmented separately and removed. Where boundaries were difficult to discern in broken sections, reliable annotation was ensured by observing unambiguous adjacent sections. We created a section tracker on Google Sheets to assign segmentations to our team, and each member was then responsible for updating the status of the cerebellum and cerebrum sections once they were completed. At least one round of quality control per section was carried out to improve annotations and identify potential problems.
The main tools we employed were MicroDraw, allowing us to annotate the histological sections, Mattermost, providing an easily accessible platform in communication and file sharing, and Google Collaboratory, enabling us to analyze our data and present our research in an organized manner. Below you can see an image of all the annotations made from the gorilla brain sections. Using MicroDraw, a three-dimensional reconstruction of the cerebellum was created in order to aid visualization of the cerebellum, produced by taking each annotation of the gorilla cerebellum, stacking them, and using a marching cubes algorithm to make the surface between each section. You can see our prototype of the reconstruction with the gorilla below. Within Google Collaboratory, the sum of all perimeters was taken and multiplied by the width of each section and the distance between them. To this value, a correction factor of 1.87 was applied, which represents the fresh brain to fixed brain volume ratio for this specific gorilla. This value was provided by Carol MacLeod. Our main findings are that the gorilla cerebellum surface area constituted 64.4% of its neocortex and a cerebellar volume of 14.3% of its whole brain. This volume measurement is comparable to MacLeod et al.'s gorilla value of 14.8% relative volume. Recalling Serino et al.'s findings, we can identify notably reduced surface area in comparison to human 78%, but nevertheless higher relative cerebellar surface area compared to macaques 33%. Therefore, as hypothesized, Hominoidea cerebella continue to possess higher surface area than non-Hominoidea.
Investigating the extent to which humans deviate from the non-human anthropoid trend, we conducted an exploratory phylogenetic analysis utilizing phylogenetic generalized least squares, with 7 hominoidea and 15 non-hominoidea. Our gorilla had 43% more surface area than expected of non-human anthropoid cerebral volumes. From this phylogenetic analysis, we found that human cerebella had 19.3% more surface area relative to cerebral volume than non-human anthropoids. And in the graph on the right, human cerebella were 15.1% less voluminous relative to cerebellar surface area. The conclusions we drew from the research are that human cerebella are less voluminous than those of other hominoidea, hominoid cerebella remain relatively more voluminous than other anthropoids, and hominoid cerebella have higher relative surface area than other anthropoids. More species will be included in the future to facilitate closer examination of cerebellar surface area relative to the neocortex, such as chimpanzees and bonobos. This could potentially provide insight into the reason for enhanced human behavior such as advanced language and tool use. Thank you for listening!